In the absence of co-stimulation, tolerance is induced. Activation of naive T cells requires interaction with cells expressing both the appropriate peptide MHC complex and co-stimulatory molecules. In the absence of full co-stimulation, specific antigen recognition leads to partial T cell activation, leading to T cell anergy or deletion. Tissue cells are not known to express B7 or other co-stimulatory molecules and can therefore induce tolerance. Experiments with transgenes show that the expression of foreign antigens in peripheral tissues can, in some cases, induce tolerance, whereas in other cases the foreign antigen seems not to be presented to naive T cells at a sufficient level and is ignored. Autoimmunity can be induced by co-expression of a foreign antigen and B7 in the same target tissue, but as B7 expression on peripheral tissue cells is not by itself a sufficient stimulus for autoimmunity, it is clear that the loss of tolerance to self-tissue requires the co-expression of both a suitable target antigen and co-stimulatory molecules. Antigens that are unable to induce clonal anergy or deletion, but can nonetheless act as targets for effector T cells, can serve as autoantigens. These antigens are likely to be tissue-specific and relatively few. By analogy with graft rejection, it seems likely that autoimmunity is initiated when a professional antigen-presenting cell picks up a tissue-specific autoantigen and migrates to the regional lymph node where it is induced to express co-stimulatory activity. Once an autoantigen is expressed on a cell with co-stimulatory potential, naive, ignorant T cells specific for the autoantigen can become activated and can home to the tissues where they interact with their target antigens. At this stage, the absence of co-stimulatory molecules on tissue cells that present the autoantigen can again limit the response. Armed effector T cells kill only a limited number of antigen-expressing tissue cells if these lack co-stimulatory activity. After killing a few targets, the effector cell dies. Thus, not only can responses not be initiated in the absence of co-stimulatory activity, they also cannot be sustained. And that's in the absence of co-stimulation, tolerance is induced. Have a great day.